Thank you for joining today's presentation. During today's presentation, we're going to talk about the transformational platform that transcends the limits of the legacy protein production technologies. The speed at which we can program these cells, the scales which we can grow these cells at, the versatility, the simplicity, the low-cost media, all those factors plus more highlight the reasons why, whether it's for an industrial product or a pharmaceutical animal health product, a growth factor, potentially metabolites, and other biological products, the C1 cells offer tremendous benefits and opportunities to help grow your business and to bring biologic products to market in larger volumes more affordably. The C1 protein production platform is being leveraged on a global basis to help speed the development, lower the cost, and improve the performance of biologic vaccines, drugs, and other bioproducts. The C1 technology is not only ideally suited to help meet the global demand for health equity, including speeding the development, improving the yields, and lowering the cost of multivalent vaccines and antibody cocktails, but the C1 cells are also applicable to a wide and growing number of other applications, both in animal and human health and in industrial biotechnology. We believe, as do a growing number of scientists in academia, governmental agencies, and in industry, that the C1 technology offers unique opportunities for us to work together with your company to develop larger amounts of lower cost products. What are the properties of the C1 cell line that provides the competitive advantages to help disrupt the biomanufacturing of vaccine, drugs, and other biologic products? The purity, the productivity, the robustness, the versatility, the speed, the flexible commercial scales, and the cost of goods, the low-cost media. Let's highlight a few of those advantages. When we compare to E. coli, where the protein is produced traditionally intracellularly, C1 secretes the protein in the media. We also do not have any endotoxins to remove from the media as well. When you compare it to CHO cells, we have a 2-hour doubling time compared to approximately 24 hours for CHO cells. We can produce 3 or 4 batches, and at the same time you produce 1 batch of a CHO cell. We can do so potentially at higher yields, at lower cost, without the need to do viral inactivation. And when you want to compare it to insect cells for antigen production, one example is when we produce the SPV antigen at 300 times greater yield at lower cost and less time than the same SPV antigen produced in insect cells or baculovirus cells. So you can see that the C1 technology platform has a robust, versatile, unique applicability across diverse opportunities. Over the past 25 years, the C1 cells have evolved from a highly filamentous nature to a non-filamentous nature. As you can see pictured, C1 is characterized by a morphology change to a fragmented, non-sporulating mycelial phenotype. This morphology resulted in very high titers and low viscosity fermentations. In addition, we've engineered out many of the protease genes to produce the protease background that led to higher productivity and greater stability. The C1 glycan structures are atypical if you compare them to yeast, such as Saccharomyces or Pichia, or other filamentous fungi. You can see that they're much more human-like. We've now, over the last several years, glycoengineered our C1 cells using synthetic biology to produce glycoengineered cells that impart G0, G2, and G2F glycan structures. We continue to evolve and modify the C1 cells so that they have low protease activity, human-like glycan structures that can be used to produce glycoproteins like monoclonal antibodies at high yields, faster, and at lower cost. Using our C1 site-directed transformation methods, we can rapidly generate highly productive, stable C1 cell lines. The advantage is that the cell lines that we create in this 7 to 8 week process are used in your commercial scale manufacturing. It speeds the development timeline, improves the probabilities of commercialization, and lowers the cost. We can go from a stable C1 cell line within a few months right into cGMP manufacturing. The scales at which we can grow C1 are unparalleled. We used C1 for over 2 decades and grew it at up to 500,000 liter scale. The media, you could use chemically defined media or industrial media, depending on your needs. If you want to make a drug or a vaccine, you can use cGMP chemically defined media based on glucose feed, or in certain countries, if you want, you can go to sucrose. The temperatures, the pH, the robustness, the versatility, the flexibility. You can use single-use bioreactors or stainless steel tanks. C1 offers flexible scale manufacturing to meet needs for a pandemic or traditional biologic drugs, vaccines, industrial enzymes, and other biological products. This slide is a subset of the types and classes and the growing number of proteins that can be produced from C1 cell lines. 24.5 grams per liter in 7 days is remarkable. 2 to 3 grams per liter of an antigen in 96 hours is even more so. The ability for C1 to produce larger amounts in a short amount of time at flexible commercial scales is unparalleled. And remember, these proteins are secreted from the media using low-cost, chemically defined media. Let us take a quick look at some of the examples of using C1 to produce recombinant vaccines for animal health. The animal health market is rapidly growing for both companion and farm animals. We've demonstrated over the 5 years plus that we've been working with the European Union Zoonosis Anticipation Preparative Initiative, both with the SPV antigen and the RVFV Rift Valley fever antigen, and now with the SARS-CoV-2 antigen, that we were able to produce very large amounts of antigens very rapidly in very large volumes at flexible commercial scales. In the ZAPI program, as an example, with the Schmallenberg virus, SPV, we were able to produce 1.8 grams per liter of the SPV antigen in a 7-day fermentation, or approximately 300-fold higher than the insect cells produced with baculovirus. In addition, you can see that the RVFV was produced almost at the same level as you saw with the SPV. Again, record levels of productivity very stably. In addition to producing the antigens themselves, the C1 technology can be engineered to produce nanoparticle vaccines. We can do it quickly, we can do it at higher levels, and we can do it at lower cost. We were successful, which we'll show you here coming up on the animal data, in cattle, sheep, and mice at producing nanoparticle vaccines with the C1 technology that were safe, effective, and protective. This slide shows you the success of our production of the SPV antigen using C1 and coupling it to a nanoparticle in both mice and in cattle challenge studies. As you can see, all immunized mice and cattle survived the challenge infection without any clinical signs of disease. Protection was conferred even after only one immunization. Just to take a brief moment, just to highlight, Dyadic C1 gene expression platform played a key role in the success of the 5-year EU $20 million ZAPI program. As highlighted by Dr. Jean-Christophe Audonnet, C1 turned in record antigen productivity for both the SPV and RVFV antigens. The C1 gene expression platform can be used to produce recombinant vaccines quicker, faster, in larger volumes at lower cost using flexible scale manufacturing. In a relatively short period of time, approximately 2 months, we developed a C1 cell line expressing the receptor binding domain of the SARS-CoV-2 spike protein. We were able to produce that initially at approximately a gram per liter, and then we optimized the fermentation process rapidly to between 2 to 3 grams per liter in 5 days in a 22-liter fermenter. We can produce it not only at high yields, but with high purity and high stability. In addition to the SARS-CoV-2 RBD, we produced a SARS-CoV RBD with a nanoparticle, the full spike with the capability to be produced into a nanoparticle, the FC RBD, and a monoclonal antibody. In addition, as mentioned before, we've been able to produce HA, MERS, SPV antigen, RVFV, etc. As you can see here, with high purity and great stability. We hit it out of the park again, in this case with a C1 RBD spike tag without a C tag, which can be used to make nanoparticle vaccines. We were able to demonstrate 700 to 800 milligrams per liter of productivity in only 55 hours, with 99% purity, and the RBD spike tag protein itself performed very well. Let's now look at the analytics and the performance characteristics of the C1 produced RBD compared to the RBD produced from CHO cells. The binding kinetics for the C1 produced RBD were virtually identical to those of CHO cells. The C1 RBD antigen was properly folded and showed a high potential to generate immune response and protection against the SARS-CoV-2 virus. In mice studies, it was confirmed that the RBD antigen produced by C1 induced the production of neutralizing antibodies against the SARS-CoV-2 in mice. The immunogenicity and protection upon challenge in transgenic mice expressing the human ACE2 infected with the SARS-CoV-2 virus were successfully completed. This mice study demonstrated that the C1 produced RBD induced high neutralizing antibody titers. You can see here there was two approaches, a prime and a boost, which is in the middle in purple, which showed very high neutralizing antibody titers, and then adding a booster on the right in the green where the numbers exponentially increased. The C1 technology produced a highly effective antigen based on the SARS-CoV-2 receptor binding domain that has now been shown in challenge studies to be safe, effective, and protective. Using this table, we show as an example, if you could produce 3 grams per liter of a C1 RBD, where you used 20 micrograms twice as a prime and a boost, you could use one 23,000 liter fermenter in 5 days to produce enough antigen to treat 1 billion people. The C1 cells can be rapidly programmed to develop and manufacture affordable vaccines and antibodies that have the potential to be safe, effective, and protective against COVID-19 variants. We're continuing to make progress on our proprietary SARS-CoV-2 RBD vaccine candidate, DYAI-100. The cGMP drug substance has been manufactured, the toxicology studies are ongoing, and we continue to advance towards the anticipated human phase 1 clinical trial to prove safety and efficacy of antigen proteins produced from the C1 cells. In addition to the SARS-CoV-2 antigen program, we're also working with the Oslo University on influenza antigens. As mentioned previously, we worked with Sanofi and demonstrated that the C1 cell line can be used to produce hemagglutinin at higher levels at lower cost with better protection. As shown here by Sanofi Pasteur, the C1 produced hemagglutinin was safe and effective. In fact, it was more effective using 1/3 the amount that we could produce at much higher levels. C1 can be also used to produce other therapeutic proteins, including monoclonal antibodies. We can produce monoclonal antibodies in 3 or 4 batches in the same time it takes to produce 1 batch of a CHO cell at lower costs, potentially higher yields at flexible commercial scales. C1 produced antibodies have virtually the same binding capacity as CHO produced antibodies. In addition, the C1 produced antibodies have the same neutralization capacity as CHO-produced antibodies. Again, the C1 platform has the potential to replace conventional mAb manufacturing by providing lower cost, faster production, and higher yields. Let's work together to overcome global health access, affordability, and equity challenges. Together, we can make a difference.